Recently you shared some really interesting stuff with us about all the ways doctors test for that thing called HER2 in cancer.
Right.
And well, honestly, it kind of got us thinking, like, wow, we really need to do a deep dive into this.
Yeah.
So that's what we're going to do today. We're going to get to the bottom of these HER2 testing methods. How do they actually work? What are their strengths? But just as important, where do they, you know, kind of fall short?
Hmm.
So by the end, you should have a way better understanding of how we figure out HER2 status and why that even matters,
Yeah,
right?
absolutely. And, a patient's HER2 status is super important because it
Well,
really determines if these uh, really powerful targeted therapies are going to work,
right.
like trastuzumab,
Yeah.
pertuzumab,
Those are
and even trastuzumab deruxtecan,
mm,
or what they call HER2.
in here too.
Yes. So understanding this whole testing thing is really, really important for personalized treatment.
Okay.
And today, we're going to focus on 5 main methods.
5. Yeah.
Yeah. First off, immunohistochemistry.
Yeah.
Or IHC.
Okay.
Then we got in situ hybridization, or ISH,
Yeah.
which actually has a few variations within it.
Okay.
Next generation sequencing, or NGS.
NGS.
Then we have digital PCR, that's ddPCR.
Okay.
And lastly, the super cutting edge AI-assisted digital pathology.
All right, so let's dive right into IHC. It seems like this one's very direct, very visual.
It is.
So walk us through the basics, how it works.
Sure. Think of IHC as a way to directly see if there's a ton or an overexpression of HER2 protein
Okay.
on the surface of cancer cells. So it uses antibodies. You can think of them as tiny little keys.
Right.
And these keys bind to HER2. And these antibodies have something attached to them that makes a color
Oh, wow.
when they bind.
I see.
You can actually see it under a microscope. And the intensity of the color tells you how much HER2 is there.
Okay, so that brings us to the scores, right, that we saw.
Exactly.
0, 1 plus 2 plus 3 plus. Where does that all fit in?
So if the score is a 0 or a 1 plus, that means the tumor is HER2 negative. And if it's a 3 plus, that's HER2 positive.
Okay.
And those are the patients who benefit most from those targeted therapies we talked about.
Okay.
But you got that 2 plus score. That's the tricky one. It's like the middle ground.
Right.
Like the information you shared said, if you get a 2 plus, you almost always need more testing with one of the ISH methods.
Gotcha. So what are the, I guess, the good things about IHC? Like, why is it usually the first test they do?
So first of all, it's available pretty much everywhere.
That makes sense.
Most labs can do it. So it's quick and easy to get results.
Right.
And it's definitely cheaper than some of the fancier molecular tests we'll talk about.
Okay.
And as you mentioned, a pathologist can actually look at it and see stuff.
Right.
They can see how much protein there is, but they can also see the pattern of how it's spread out in the cells.
Interesting.
Sometimes that can give you like little hints for diagnosis.
Wow. Okay, so that's the good side. What about the bad side? Like, what are the problems or challenges with doing it this way?
The main problem with IHC is that scoring can be subjective.
Okay.
One pathologist might see a strong 2 plus. Another might say it's closer to a 3 plus.
I see.
It's what we call inter-observer variability. That's why that 2 plus result always needs more testing.
Right. Right.
And, the information you shared also mentioned tumor heterogeneity.
Yeah.
That's a big one. A tumor isn't always the same throughout.
Right.
Some parts might have lots of HER2, others might have very little. So if you take a small sample, you might miss the parts with high HER2.
I see.
And then you might underestimate the HER2 status.
So that heterogeneity thing, is that like a problem for all these tests to some degree?
Yeah, it is. Because, you're only looking at a tiny piece of a much bigger tumor.
Right. Right.
Which can be very complex.
Interesting. Okay. So let's move on to in situ hybridization, or ISH. And this is where instead of looking at the protein, we look at the genes themselves, right?
Right.
So what did the information you reviewed say about how ISH works?
So instead of just looking for extra HER2 protein, ISH actually looks for extra copies of the HER2 gene in the DNA.
Okay.
It's called amplification.
Yeah.
And there are a few ways to do it, like FISH, CISH, and SCIH.
Wow, lots of acronyms.
Yeah. So FISH stands for fluorescence in situ hybridization.
Okay.
And it uses these special probes that are fluorescent and bind to the HER2 gene. So if a cell has multiple copies of the gene, you'll see more fluorescent signals.
Okay.
You need a special microscope to see it, though. FISH is often considered the gold standard for finding HER2 gene amplification.
Oh, I see.
But then there's CISH and CSIH.
Right. So what's the difference between those and FISH?
So both CIH, which is chromogenic in situ hybridization,
Mm-hmm.
and SAH. silver in situ hybridization, do the same thing as fish. They look for HER2 gene amplification. But they use different ways to see it. Instead of glowing, they use chemical reactions that make either a color or silver particles where the probes bind.
Interesting.
So you can see them with a regular microscope,
Mm-hmm.
the kind most labs already have.
Right.
And a big plus for CISH and SIH is that the slides are permanent, like IHC slides, so you can store them and look at them later.
I see. So the end goal is the same, like seeing if the gene's amplified.
Mm,
But the way you see it and whether the slides are permanent, that's the difference, right?
exactly.
So what are the good things about looking directly at the genes with ISH compared to just the protein levels with IHC?
The big advantage is that ISH is much better at measuring gene amplification.
Okay.
You actually count the HER2 gene copies and compare them to control genes. So it's more objective
Right.
compared to just eyeballing the protein levels with IHC.
Right.
This is really helpful for those borderline cases where the IUC is a 2+,
Right, where it's not clear.
and you just don't know for sure.
Okay, makes sense. And what are the problems or challenges with using ISH? It sounds like there are some.
Yeah. So a FUSH can be kind of tricky and usually more expensive.
Why is that?
You need special microscopes and stuff.
Okay,
And all the ISH methods need a certain level of, skill to read the results.
makes sense.
With FUSH, sometimes the signals from the genes are super close together or even overlap, so it can be hard to count them accurately.
Oh, wow.
And while CISH and SEH here are easier because you can use a regular microscope and the slides are permanent, you still need a really good pathologist to look at them.
Okay, so we've covered the visual protein check with IHC
Right.
and the gene amplification check with ISH.
Right.
Now, the information we got also mentioned next generation sequencing, NGS.
Yes.
And this one sounds like it takes things to a whole new level, like really understanding the genetics.
Oh, it definitely does. So IHC just gives you a snapshot of the protein,
Right.
and ISH looks at how many copies of the gene there are.
Mm-hmm.
But NGS actually looks at the DNA sequence itself.
Wow.
So you can analyze huge chunks of the tumor's DNA.
That's way more information.
Way more. You can see if the HER2 gene is amplified, but you can also see if there are any mutations in the gene itself.
Oh, wow.
And you can see changes in other genes that might be important for how the cancer grows and behaves.
So it's like a whole genetic blueprint, not just HER2.
Exactly. And this is a huge advantage. Knowing about other genetic changes can really help you figure out the best treatment.
That makes sense.
And maybe even see if the cancer might become resistant to the HER2 targeted therapies.
Right. Right.
For example, sometimes you see co-mutations that suggest you should use a different treatment from the start.
Interesting. It also mentioned that NGS is used in liquid biopsies,
Oh, yeah.
which that still sounds like sci-fi to me.
I know, right? It's pretty cool.
So how does NGS play a role in that?
So liquid biopsies analyze circulating tumor DNA, or ctDNA.
Okay.
These are tiny bits of DNA that cancer cells shed into the blood. And NGS is so sensitive, it can analyze this ctDNA to see if the HER2 gene is amplified or mutated,
Wow.
all without having to do a biopsy.
No kidding.
Yeah. This is really helpful for keeping track of how a patient's HER2 status is changing during treatment, or even to catch a recurrence early on.
That's amazing. Less invasive, and you can like monitor the tumor over time.
Exactly.
So it all sounds pretty good. What are the downsides or limitations with NGS?
Well, the main problem is the cost.
Yeah, that makes sense.
It's way more expensive than IHC or ISH because the equipment is really fancy and the chemicals are expensive.
Right.
And because you get so much data, you need special computer programs and experts to understand it all.
Yeah, I bet.
It's like trying to find a needle in a haystack.
Wow.
And usually it takes longer to get the NGS results compared to, the standard IHC or ISH.
Okay, so higher cost, tricky data analysis, and longer wait times. Good to know. All right, what about digital PCR or ddPCR? It sounds like it also uses liquid biopsies,
It does.
but in a different way than NGS, right?
Right. So NGS with liquid biopsies gives you a really broad look at the genetics,
Okay.
but ddPCR is more focused. It looks specifically at how much the HER2 gene is amplified in the circulating tumor DNA.
So instead of just saying yes or no, like amplified or not, it gives you a number, like how much it's amplified.
Exactly. It takes the DNA and splits it into like thousands of tiny droplets.
Wow.
And then in each droplet, it copies the HER2 gene over and over again.
Oh, wow.
That's called PCR, by the way, polymerase chain reaction.
Okay.
It makes it easier to see even small amounts of DNA.
Right.
Then They count how many droplets have the amplified HER2 gene,
Okay.
and that tells you exactly how much it's amplified in the original blood sample.
So it's super sensitive. And it can see even really small amounts of HER2 DNA.
Yep. So it's really good for tracking minimal residual disease after treatment
Okay.
or catching a recurrence early.
Right, right.
And because it's all done in the blood, it's non-invasive.
Less needles. That's always good.
Exactly. And you can easily monitor how the HER2 levels are changing over time.
Awesome. But again, there must be some things to watch out for with ddPCR, right?
Yeah.
Yeah.
Because it's looking at DNA in the blood, it might not perfectly represent what's happening in the main tumor.
Okay.
Remember that heterogeneity we talked about?
Right.
If the tumor doesn't shed DNA evenly into the blood, the results might not be accurate for the whole tumor.
Makes sense.
And even though ddPCR is really promising, they're still working on standardizing it.
Okay.
So it's not widely used yet.
Wow.
And like any liquid biopsy, the results depend on having enough good quality ctDNA in the blood sample.
Gotcha. So it's a really precise tool for a very specific thing. But you got to keep those limitations in mind.
Exactly.
Okay, the last one on the list, AI-assisted digital pathology. Now, this sounds like the future, like computers reading our tests.
I know, right? It's super exciting.
So how does AI actually help with HER2 testing?
So it uses machine learning algorithms to analyze images of IHC slides. Basically, you feed the computer tons of images that have already been scored by pathologists.
Right.
And the AI learns to recognize patterns and little details that correspond to each score. 0, 1 plus, 2 plus, 3 plus.
So you're teaching a computer to read the slides like a pathologist would.
Yeah, exactly. The goal is to have AI score the slides automatically, or at least help the pathologist do it.
Wow.
One of the best things about it is that it could make things way more consistent.
Okay. How so?
Well, you take out the human subjectivity, so you wouldn't have different pathologists giving different scores for the same slide.
That makes a lot of sense.
That would mean patients get more accurate and consistent results no matter where they're being tested.
Right, right.
Another big plus is speed.
Okay.
AI can analyze images way faster than a human can.
Right.
So it could really speed things up in busy labs.
Right.
And imagine we could finally have true standardization in IHC.
That would be huge.
It's been a problem for so long. Yeah. But there must be some pretty big challenges to, making this work in such a complex area.
Oh, definitely. The AI is only as good as the data it learns from.
That makes sense.
If the images aren't good quality, or if they don't include enough variety of tumors and staining patterns, the AI might make mistakes when it sees a real patient sample.
Right.
And a lot of these AI models are still being developed and tested.
Okay.
They need to be really rigorously checked and approved before they can be used on patients.
Yeah.
And of course, you need the right equipment,
Like what?
like special digital systems to get those high-res images.
Right.
And the AI software itself, people who know how to use it.
So it sounds like AI has the potential to be amazing,
It does.
but we need to be careful about the data quality, really validate it, and figure out how to actually use it in real life.
Exactly.
So we've covered 5 different HER2 testing methods.
Yeah.
What's the main takeaway for you?
I'm curious to hear your thoughts. Well, it's clear that getting this HER2 testing right is super important for deciding on the right treatment.
Definitely.
And even though those older methods like IHC and ISH are still really useful and easy to access, these newer technologies like the NGS, ddPCR, and the AI, they're giving us so much more information,
Right.
especially in those tricky cases where IHC or ISH just aren't clear enough.
I agree.
Yeah, it's like they're giving us a deeper understanding, more precise measurements in certain situations.
Yeah, there is especially helpful when you have that heterogeneity where the HER2 is different in different parts of the tumor.
Right.
Or when the HER2 levels are really
Yeah.
low,
Right.
they can give you a much clearer picture.
And it seems like everyone's working hard to make all these methods better, more standardized, more accurate, so they can really help patients.
Oh, absolutely. The field's constantly changing.
Yeah,
Researchers and doctors are working hard to, overcome the limitations we talked about
right.
to make each method as useful as possible.
This has been a really cool deep dive into this whole HER2 testing thing.
I agree.
It's amazing to see all the science and technology that goes into, just figuring out this one aspect of cancer.
It is. Each method has its own strengths and weaknesses, and understanding those is key to, really appreciating how far we've come.
Absolutely. So with all these different methods out there, each with its own good and bad sides, How do you think HER2 testing will change cancer treatment in the future?
Right.
It really makes you think about, how easy it is to get the tests, how much they cost, and how much information they actually give you,
Yeah,
and how all those things will shape, the future of cancer treatment.
absolutely. It's definitely something to keep an eye on.
Yeah,
It's a really exciting time for this field.
it is.